Hey guys, this video is all about passive transport, which is one of the very many functions of the cell membrane. Just remember that the main function of the cell membrane is transporting goods in and out of our lovely cell, and it is this picture just shows a little tiny chunk that cell membrane will actually span all the way around the cell and will be acting as a sort of border to control what's going to come in and out. There are a couple different types of passive transport, and they have a few things in common. One, they do not require any energy or ATP, and it also always involves a molecule traveling from high concentration to low concentration. Let's say, for instance, there's a high concentration of a molecule outside the cell here. A lot of times when we use the word concentration, it's referring to the amount of something. So in this case, high concentration is just a fancy way of saying, oh, there's a lot of green dots outside the cell right now, floating around in the what's called the extracellular matrix, which is the space between two cells, or the space in the outer part of our cell that we're looking at right now. So let's say this green molecule wants to travel inside the cell from high to low. That just means there's going to be less green dots on the inside of the cell in the cytosol or the cytoplasm. Now, how this molecule actually passively transports into the cell is going to be determined by if it's a polar, hydrophilic, water-loving molecule, or if it's a nonpolar, hydrophobic, not really into water molecule. And that's just going to determine how it flows back and forth. And that is going to bring me to our two major examples of passive transport. Diffusion is a really general term that we use to describe how molecules travel through space from high to low. And that is going to apply here in the cell. And it's also going to apply when you put food coloring in a glass of water and it naturally spreads through the water and will kind of turn one uniform color over time. It also sort of describes how if you were to spray body spray, it's going to go from high concentration right out of the bottle where you sprayed it to low, aka maybe in a room you're in where people who are close to you will smell it first and then further and further away. The concept is very similar to our cell. It's just saying that, hey, these molecules are going to pass from one side of this membrane to the other from high to low, pretty much no matter what. And diffusion is going to refer to molecules that are nonpolar and can pass right through the middle of our bilayer. So in this case, our little green substance is going to be traveling from outside the cell to in, from high to low, right through the middle of the bilayer. Because if you remember, the bilayer is where the fatty acid tails of our phospholipids, or the lipid part of the phospholipids, are located. And so the middle, where all the yellow lines are in this drawing, is going to be hydrophobic. So other hydrophobic molecules can easily pass through that particular region. For instance, this particular molecule can travel directly through to the other side or from out to in. So what about all of the polar molecules? So let's say that our green molecule is actually polar here. Well, we know that polar molecules are hydrophilic, meaning they are water-loving. Well, if there's one thing we also know about polar molecules, it's that hydrophilic things do not mix with nonpolar hydrophobic things. So if this molecule was polar and tried to go into the membrane, it's going to get rejected back out because it literally can't mix with that hydrophobic center. So these molecules, they just can't pass through the bilayer directly. So they have to resort to using our lovely transport protein, which has been chilling in the middle of this drawing, this entire video. This blue guy right here, he's a transport protein. And they are happy to help our lovely polar molecules get into the cell. So a polar molecule pretty much uses the transport protein like a bridge and will travel through it from high to low still. That's the key. It's still passive, no energy, it's high to low, but it will pass through that transport protein to the other side of the cell. This works really well because our transport protein is actually hydrophilic, just like our polar molecules, which makes them mesh really well and work well as a team. So the next thing we're really going to talk about is that super important polar molecule that we've been harping on all year, which is going to be our lovely, lovely H2O. Water is so important that its particular facilitated diffusion gets its own name, which is osmosis. And osmosis, because water has to use a transport protein to get in, is still passive. So water still always moves from high to low. And sometimes that involves water being more concentrated outside the cell than in. So when water is super concentrated outside the cell in comparison to inside, so usually, a cell is going to be about 70% water, meaning that 30% of the cell is going to be like your organelles, your salts, your sugars, various molecules floating around that aren't water. That's normal. 30% of your cells are typically going to make up something besides water, because if you look in the mirror, you're not just staring at a giant water blob, you have other parts to you. So think you're not 100% water, so neither is the inside of the cell. That kind of helps me visualize what's happening. So on the left-hand side drawing, you can see that this particular cell has been placed most likely in a solution that is 100% pure water, because look at all those water molecules. And on the inside, we have about 70% water. So what's going to happen is water is going to naturally pass through the aquaporin, or the transport protein for water, into the cell. It doesn't matter if the cell wants more water or not, it's going to happen. So a lot of times in these extreme situations, this can actually cause the cell to explode because it fills up with water so much that it actually breaks and bursts the cell membrane, kind of like a water balloon that has been filled up with too much water. So that particular solution is called hypotonic, meaning there is more water and less solute. Solute being fancy for something not water. You could pick anything, salt, whatever. So I always say, remember hippo, because hippos are kind of large and round, and hypotonic solutions cause the cell to swell up kind of like a balloon and possibly burst. So let's look at the flip side of this particular situation. So now here on the right, we have a situation where we have our cell, 70% water, and we place it into a solution that is super high in some sort of solute, so its water concentration's gone down. So think like the more sugar in a solution, the less water there is, just because that's how space works. So in this particular case, the water, because it's always going to travel from high to low, is going to go from inside of our lovely cell to the outside. And this again happens no matter what. So your cells, your body doesn't want to get dehydrated, but if you go chug a bottle of soy sauce, all that sodium is going to cause water to rush out of your cells to balance it out, and it is actually quite dangerous. So this would be a hypertonic solution, which means that there's less water and more solute, or sugars and salts, things like that. And this is really going to cause your cell to get dehydrated and shrink and lose water. But what if the solution of water is the same on the outside and the inside? So say you put a cell in 70% water solution, meaning the water is going to be the same on both the inside and the outside. So in this case, water is still going to move from one side to the other. It's just going to be more at an equal rate. Now, if that confuses you, try to think of some solution that's hypotonic or pure water. Because there are physically more molecules of water to interact with the membrane, the chances of them flowing from the really watery, pure water solution into the cell is higher. So thus there's going to be a net movement into the cell, and vice versa for your hypertonic, where the water is actually going to leave the cell. So in this case, because it's pretty equal, water is going to flow back and forth from outside the cell to in, and from inside the cell to out at an equal rate. So in this case, there is going to be no net change. We refer to these types of solutions as isotonic. It's important to note that this means that there is an equal amount of solute, sugars, salts, and things, and water, which is going to lead to a no net change or percent change in mass. This is exactly what we saw in our experiment when we were looking for that 0% change to figure out how much solute was in the cell. The reason why we were looking for 0% is because that is where you can find how much solute is in the cell in comparison to the solution it's in. If you put a cell in a 0.4 molar sucrose solution and it doesn't really lose water, that tells you that that cell probably has around a 0.4 sucrose concentration as well. So here we have 3 sample problems, pretty similar to what you've seen in class with me before. So this would be a good time to pause the video if you want to test yourself, or you can just listen to me explain it. As an example, I'm going to show the arrow of water movement, the percent change in mass that that particular cell would experience in that solution, and what type of solution that is. So if you don't want to hear the answers yet, you should pause the video now. Okay, let's look at this sample A. So you have a 0.1 molar sucrose solution on the inside of the cell and a 0.8 molar sucrose solution on the outside of the cell. So in this case, there is a lot of sugar in this solution. So 0.8 is really high. 0.8 is higher than 0.1. So one thing you need to remember is that the more solute, the less water. So that particular solution type is going to be hypertonic. And you can actually go back to the definition and look and see that that is indeed defined by low water, high solute. Because the more solute in the solution, the less water. So in this case, water is going to go from inside the cell to out. And that is going to lead to a negative percent change. And this is hypertonic. For sample B, this particular sample has a 70% water solution inside the cell and a 30% salt solution outside the cell. So if there's 30% salt outside the cell, it's pretty safe to say that that's also means that there's going to be 70% water, since we're going to assume that this salt is whatever isn't salt in this solution is water. So you can see that the water concentration here is actually equal. So I'm going to draw an arrow pointing both in and out. This is going to have a 0% change. So we can actually assume that our cell has about 30% salt or 30% solute, and this solution type is going to be isotonic because it's equal on either side. And lastly, we have solution C, where you have a 0.8 molar sucrose solution on the inside of the cell, and we have a 0.2 molar sucrose solution on the outside. So in this case, again, kind of like sample problem A, the more sucrose you have, the less water. So our cell actually has a lot of sugar in it. So maybe this is a sweet potato cell. We saw that the sweet potato data had about 8 molar sucrose. So perhaps we put a sweet potato cell in a 0.2 molar sucrose solution, which Some of you did. And what happened was, because there's not really a lot of water in that cell in comparison to the outside, since the more sugar, the less water. And in this case, the outside has less sugar, meaning more water. You would experience a situation where water is going to move into the cell. So you would expect a positive percent change, meaning that that cell is going to gain water, it's going to gain mass. And the solution type for sample problem C would be hypotonic, meaning that there's more water outside the cell than inside the cell. So water goes in. Just a reminder that what all three of these have in common is that water always moves from high to low passively without the use of energy. And that wraps up our passive transport video.